I'm Morgan Hawkins. I'm a PhD student here at the Marine Lab. This is my 4th year, and the last year I've been working on a aquaculture project focusing on the effect of probiotics with the larval culture of eastern oysters. This project was funded by the Aquaculture Review Council, or ARC, which is directly under FDAX. So why is this important? Well, hatcheries worldwide and specifically in Florida face bacterial issues when rearing larval oysters in hatcheries. And these are a couple different examples of some of those bacterial issues you can find while you're culturing oysters. It can take place in the digestive tract or in the velum, which is an organ responsible for swimming, breathing, and eating while they're in their larval stage. It can also cause detrimental mortality to newly set seed. So this is very important to a hatchery that can affect your overall production if your culture becomes infected with a bacterial infection. A common example of one of these pathogenic bacteria is Vibrio. You've probably heard of this quite a lot. In a hatchery setting, Vibrio can remain undetectable for the first couple days of culture, making it crucial for hatcheries to actually prevent this from happening because there's not a good way of correcting it. So once you get into these later stages, the hatchery can kind of find out that they're undergoing a bacterial infection by some malformities in the velum, that organ that I talked about earlier. You can kind of see that it starts to clump and fall off and ultimately leads to production death. So again, it's important to understand why these mortalities are happening, how to prevent them in order for hatcheries to upkeep their seed production and sell seed to you guys. So a method of hopefully treating this issue of bacterial infection is prevention. So again, like I said, these bacterial infections are likely undetectable during the first couple days of culture. So really, we are dedicated to preventing this from even starting versus actually correcting the issue. So a prevention method that is common in other aquaculture industries is using probiotics. There's a couple different methods or mechanisms that the probiotic can help larval cultures. One is that the probiotic itself, which is just basically beneficial bacteria added to the water, can outcompete harmful bacteria. So then that way you're establishing a more healthy microbiome for your larvae to grow in. Another mechanism to which that it could assist in larval growth and survival is the development of immunity. So these larvae ingest the probiotic and actually can better fight off bacterial infections, viral infections, and parasitic infections. And then lastly, another mechanism that I'll highlight here is the enhancement of growth and survival. So the idea here is that these larvae are actually ingesting the probiotic. The probiotic is working just how we would assume like a probiotic would work for our digestive system. It colonizes the intestines and actually helps them digest food and make energy. And so therefore you get this associated enhanced growth and survival from that extra digestion. So this is very common in shrimp aquaculture. As you can see from this graph, there's a lot of different publications for the use of probiotics in culture. So that's why it's important to understand if these probiotics can also work for shellfish. So, and also this method of prevention is an alternative to antibiotics, which we can't actually use for farm-raised animals, and phage therapy, which requires a lot of extra time and research to actually make a bacteria that eats the harmful bacteria. So this project has a bunch of objectives, but the first objective overall is just to determine whether or not the larval survival and growth is enhanced with the use of a probiotic. And we also tested this seasonally, so running the spawn during the fall and during the spring to see if that changes with the seasonality. We also wanted to discover if this probiotic, which the name is Cyano-like MIC, it's actually available online through IV Aquaculture, alters settlement success. So the idea is if these larvae are growing rapidly and they're more healthy than normal, then maybe they can have a better metamorphosis success and become seed. And then another objective for inside the hatchery is to determine whether or not you should use a high probiotic concentration or a half dose low probiotic concentration. And that's just because Again, these probiotics aren't common in shellfish aquaculture yet, and so a lot of the literature is based on shrimp aquaculture. So we want to identify which concentration would work best for the oysters. On top of this project, outside of the hatchery, we actually implemented some sequencing techniques to better understand what the native bacteria are and how they change seasonally. So a couple of objectives here for those sequencing techniques is one, to just identify the species that's specifically in this probiotic. Two is to determine if this probiotic actually changes the relative abundances of other bacteria while inside these tanks. And three, just determine how the microbial composition changes naturally during the seasons. So the methods here, we implemented commercial stocking density. So all of the methods is copying basically what goes on in a typical commercial hatchery. So this makes it super applicable to any hatchery working on the Gulf of Mexico, just to ensure that our methods match up with commercial stocking densities, live feeds, daily water changes, and again, repeating this for fall and spring. And then again, to highlight the Illumina sequencing, we worked with the Atmospheric Science Building at FSU for library preparation and sequencing. And this method will allow a full sweep of bacterial, fungal, and archaea composition compared to other methods. So this would be interesting. And I have some results to share with you today. I'm actually ending the spring oyster experiment right after this meeting, so I don't have the specific graphs for that, but I have some cool data from the fall experiment and some cool data from the larval spring experiment. And the sequencing has not been, the data hasn't been sent to me yet, but they are currently being sequenced at UConn at the moment. So right off, I just wanted to provide you guys with a visual. So this is taken on the same day, where these are oysters raised in a control treatment, so no probiotic, and these are oysters raised in a high probiotic situation. And just visually, you can tell that there's a huge boost in growth. You can also see in these cups, these two cups here on the left are controls, so no probiotic. These two here, one is a low probiotic, so half the dose of the high probiotic, which is here. And just telling by how dense these cups are, you can kind of get a feel for the survival being different as well. So here are some fun statistics for our fall run. So to start off with the control, we're going to first talk on larval survival. So during the 15 days of larval culture, how many did we survive? How many made it through that stage? For our control, we had 30% actually make it through the entire 15 days. Whereas the high probiotic, we had 68% of them actually make it until setting. And in our low probiotics, around the same, around 67%. Now moving into percent competency. So this is larvae that are showing signs that they are ready to metamorphose. They have an eye spot, they have a foot, and they're also grading out on 220 microscope. Here we had all of these 30% ended up having 66% of them being competent. So 66% of those guys are ready to become seed. Around the same for the high probiotic with 68%, and around a little bit more actually for the low probiotic with around 75%. And then lastly, just calculate metamorphosis success. So after these competent larvae have settled, metamorphosed, and given some time to outgrow any unsuccessful metamorphosed larvae, then I could calculate metamorphosis success. So out of here, we had 80% of those competent larvae remain alive and ready to grow, 73% in the high probiotic treatment, and 71% in the low probiotic treatment. So just to kind of get at the level of competency for the larvae dependent on control, high probiotic, low probiotic, even though we saw the same amount of being competent, this is a great graph that shows you how many actually got to competency. So when comparing the control to the high and the low, we increased our yield almost double because of using this probiotic. So again, directly impacts seed production for hatcheries, increasing the amount that we can make. And then over time, I monitored post-metamorphosis just to see if those shellfish exposed to a probiotic maybe had an overall enhancement of growth after metamorphosis. So I monitored them for 14 days after that period, and between the control, which is this first bar graph, the high, which is this middle, and the low, you can see that the size distributions are roughly the same. So they really had the same amount of growth afterwards. So once they were done metamorphosing and initially eating and living in this probiotic water, the effects kind of wore off. So spring, or I'm going to go through all the same statistics again, but I'm going to say that they're still in progress. So I don't have any of the ending kind of finale results yet. But percent survival for the control was only 14%. In the spring, we start to see high bacterial loads in our water, so that's going to decrease a little bit. But the high probiotics still outperformed double compared to the control by having 35% still remaining, and low probiotic around the same, around 30%. And when we get to the percent competency during this study, we saw 50% of those 14% larvae actually becoming competent. And in the high probiotic, we actually saw 70% of those actually still be competent. So it seems like this probiotic is actually helping during the development of becoming ready to metamorphose during a stressful environment like the spring high load bacterial load in our hatchery. And then low probiotic was around the same as the control, around 54%. And then lastly, for the metamorphosis success, 32% for the control, 24% for the high probiotic, and 27% from the low probiotic. So really no difference there. So take-home messages. It seems like our probiotic study has promising results for our larval oysters and has the potential to increase seed production in the Gulf of Mexico for producing seed for farms around the county. Seasonality does seem to play an important role for the overall effect of the probiotic. So obviously the survival for the spring trials was lower, regardless if you use the high probiotic or low probiotic controls, but it does seem to overall always outperform the control on the terms of survival. And then lastly, high probiotic concentrations seem to be necessary during spring runs when you are dealing with this high bacterial load. But if you're concerned with saving money, you can use low concentrations during the fall if you're not experiencing a bacterial load. And that is all I have for you guys. And I take any questions. Are we saving questions for the end?
No, go right ahead.
Okay.
I think that's really exciting information.
Pretty cool. Yeah. Thank you. Yeah.
What is the, so often we'll hear from hatcheries like, we're not succeeding. What is that happening at these times typically? That's the.
Yeah, it's really common for hatcheries to really not know why poultures aren't making it. And so a lot of what it gets blamed on is either bacterial issues, food quality issues, and that can all stem from water quality issues. And so this was something that we discovered here. You know, the picture I first showed with the red staining, we were like, it's probably a bacterial issue. And so that's when we started investigating the probiotic.
Can we test our growth to see, to know what we need to do?
For the use of a probiotic. So there's a lot of different methods that you can test your water with. A common method is actually plating. And so you can use auger plates and kind of swipe some seawater and you can see what bacterial communities come up. So hatcheries can use that just to determine if they're having a high bacterial load season or a low bacterial load season. But this new Illumina sequencing technique that I just brought up for this study is actually, in my opinion, an easier way to identify a more wide range of bacteria that could be present in water. Yeah.
Are hatcheries using probiotics now?
I believe it's starting to become more and more popular, and there's definitely a lot more work being done at BIMS and Oregon State looking at a true oyster probiotic that's actually sourced from the adult oyster's digestive system. And so they can cultivate that and implement it in the hatchery. Now, the only issue with that area is that it takes a lot of work, right, to actually isolate that bacteria that's beneficial. But they've had quite good success with introducing that bacteria and those larvae outperforming controls when exposed to Vibrio.
There's very few commercial probiotics available now, and yes, it tends to be more related to species specific. You mentioned University of Connecticut's doing a lot of work in conjunction with NOAA, their Milford Shellfish Lab.
Yeah.
Yeah.
And I'd like to add that the study here used a probiotic that's relatively inexpensive. It's only $150 per jar. And that jar, I haven't even used the whole thing yet, and I have done countless spawns using it. So it's inexpensive. It seems like it's increasing our yield and making our larvae more happy and healthy.
Alright, so is that one just made specifically for Eastern oyster, or is that one pretty for any?
Yeah, this is a generic probiotic. And it's marketed towards shrimp hatcheries, but it actually uses the same species of probiotic that has been shown to enhance larval oyster growth. And that's why I selected it.
Okay, wonderful. Good work.
You have a question on Zoom. What are the benefits for grow out?
For grow out, that's what I was trying to detect with monitoring them 14 days post metamorphosis. However, I did not see any long lasting effects of using the probiotic. So I would say that here, the benefit to all of you is that your hatchery might have more reliable and more quantity seed production.